From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 89, recorded on April 19th, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey there,
Nels.
Vincent. Great to be back with you, live streaming again. I've got, up front here, I'm doing some personal virology or microbiology. I've got a little head cold, and so I've got some tea. I've got an emergency bag of throat lozenges. There's a small chance I'll have to throw on my mute button for a moment while I make my way, navigate a coughing fit. So hang in there if that happens. But actually, last week I was giving a seminar and ended up losing my voice in the middle of it, which if you're giving a talk actually about the evolution of viruses and infectious microbes, I think there's something about really bringing it front and center all the way into your vocal cords somehow. But anyway, that's where I'm coming from. Hopefully I'll be able to make it through here. How are you doing, Vincent?
Well, I'm recovering from TWiV 1000.
Yeah. Wow.
Oh my gosh.
Big event.
Oh my gosh. And just the leading up to it, boy, what a lot of work that was, and so much worry and so forth that everything would go well. And, we had some technical difficulties with the audio in the theater. We started 20 minutes late, but it all went off perfectly. We had a full house, 130 seats, 130 seats sold, and it went without a hitch. We had a great bunch of things we did. I came out and introduced everyone one by one, and then we did a little science reminiscing, and then I took some audience questions, and then we played TWiV Jeopardy at the end, which was really, really a lot of fun. Oh my gosh, it was so much fun. We I made up some questions with Brianne. She got up there and was the moderator because I said to her, "Hey, Brianne, I heard you were on Jeopardy recently. Tell me how that went." And she said, "Okay, Vincent, here's how it went." So she got up there and did it, and the questions were great. So now we did a 4-camera shoot, Nels. I don't know.
Yeah, very cool.
Cameras all over the place.
Yeah,
and there's audio, 8 channels of audio. So Ray Ortega came from California. Ray used to work with me for ASM, and he still does TWiM, but live shows he still does as well. And Chris Kandian also used to be at ASM. He was the photographer for this event because thank you. Because he's become an incredible street photographer.
Wow.
And he's very famous, so I got him. He's made some incredible images. Go on Instagram or Twitter, guys. You can see them. Anyway, Ray is working on the editing.
Gotcha.
And he's probably going to not release it till later in the week. So for the first time in years, we will have the past weekend without TWiV.
That's right. Yeah, it was a rare gap in the action.
It's just really funny. But
wow.
Anyway, it was fun, and I'm happy it's over. But it's kind of a letdown, after you do a big event, right?
Oof, of course. Yeah, all the energy.
But I'll be okay. I'm busy. So tomorrow I'm going to New Orleans. Next week I'm going to New Hampshire. So it'll be all okay.
Awesome. Back in the business. Yeah. No, that's a big one, and big congrats from here. I can't wait to see it. I'm looking forward to that. I did watch Brianne on Jeopardy itself a while back. That was a really impressive turn that she had. And my goodness. So remind me, Vincent, how long has how many years has it been that TWiV's been running?
2008.
Yeah. So 20, or what is that? Do the math, 15?
Yeah, something like that. The first episode was dropped September 24th, 2008.
Okay, so coming up on 15 years this September. Yeah, and to get to 1,000 episodes in that time is really remarkable.
1,000 episodes. This is the thing that blows me away. First of all, that 130 people paid money to come, actually, as far as California, all over the country.
Cool.
California, New Mexico, Michigan, Wisconsin, New England. And they were amazing. And afterwards, we had a reception. And the place was packed. We had great food and wine, and everyone was so gracious and thankful and coming up to me and all the other hosts and talking to them. And so I was just very humbled, right, by the whole thing. And then, so we had 2 hours of reception, and people left, and it was just a blast. So I want to thank everyone who went, and I don't know if anyone here went, probably not. This is not the TWiV core, but
but we'll all be catching up when Ray releases the edited version into the wild. We'll all, many of us, myself included, I'm sure, will be tuning in.
Check it out.
Celebrating with you.
I want to shout out to a listener in Kazakhstan.
Oh wow! Yeah.
Saikal, I think is how you say it. Hello, and thanks for listening. And also from all of you, we have Andrew, our moderator. Thank you for joining us. We have Tom. We have Barb UK. Let's see who else. I think those are our moderators for today.
Great to see you all.
Who else is here? Where are you from? Nicola is from Italy. Nicola said, is Nels slowly trying to make Vincent love math? Yeah, sure.
We'll do the experiment a little bit today. We're not going to go too far under the hood of this modeling paper, but yeah, we'll spend a little time thinking about some quantitative strategies for thinking about the evolution of cancer cells, or of tumors.
Yeah. Let's see, who else we have here? Well, Andrew, of course, is from New Zealand. That's pretty cool. Tom is from Eugene, Oregon. Barb Mack is from the UK. Lori is from Tomball, Texas.
Hi, Lori. Welcome.
Not far from the, what do you call it? The science mill there.
Science mill? Yeah, I don't know where Tomball is relative to Hill Country near Johnson City, Texas. It's a big state.
Yeah, I know. It's probably very far away.
I'm not sure. Lori can let us know.
Ooh, look at this. Helsinki.
Fantastic.
Emmy
Welcome.
is from Helsinki. That's pretty cool.
Spring is arriving.
To make this chat bigger, I can't really see it. My eyes are failing. Helsinki is pretty cool. What else we have here? Dutchess County, New York. That's pretty cool. Boulder, Colorado.
Hello, Peter.
San Jose, Norfolk, England. Rome. Oh, Jan. Hello, Jan from Roma. Jan was at TWiV 1000. Yes.
Oh, very cool.
I forgot that I think she's the only one here who was there.
Crossover listener.
Let's see. Anybody else? Don't worry. It won't be long till 2000. Well, I we're doing 2 a week now, and that's 10 years. So now, am I going to be around in 10 years?
Yeah, I think you will be, Vincent. You've got that. The strength seems to only be building in terms of podcasting. But I do have to say, when you were going, weren't you going up as much as 3 a week, kind of in the heart of the pandemic? I mean, that's a lot. And so,
that's too much. We can't do 3 a week.
Yeah, correct. Yeah, and certainly, I think the pandemic and just sort of as we were grappling with that in real time, that was sort of all hands on deck moment. That's hard to sustain.
Yeah,
that kind of
But 2 a week. Elizabeth was there. Yes. Did you say hello to me, Elizabeth? If you did, I'm sorry I don't remember. If you didn't say hello, why not?
Another crossover listener. Welcome.
Cheryl is from Santa Rosa.
Cheryl.
And we have North Carolina. Slovenia. Wow, that's cool. Slovenia. We have Detroit, Michigan.
Awesome.
We have Tomball is north of Houston. I'm going to be
not too close to the hill country of West Texas.
I'm going to be in Houston in June. Yeah, doing ASM.
Oh, very cool.
And Jan says TWiV 1000 was a blast.
Nice.
Yeah, it was. It was really cool. Oh, Bangkok. I might be going to Bangkok in the fall. It's possible. All right, so that's what we have here, and that's a
Yeah,
great mixture, right?
I
That's
agree.
just a
A
great
great crew
mixture.
has already assembled, which really breathes life into the conversations that we're having here about science emerging in real time. So thank you all for being here. Really appreciate that. So Vincent, let's dive in. Um, the paper that we're going to be tackling today is open access. I saw that you put all points bulletin at the very least on Twitter to sort of tip our hand about the paper that we're going to be doing today. I'm guessing you were also out on the Discord server, hopefully, sending the link out. I guess Discord has been in the news recently for more national security related issues. I
Yeah.
don't think we have those problems with what we're doing here, all all open and public. And so, to cut to the chase, the paper that we're going to do is state dependent evolutionary models reveal modes of solid tumor growth. Uh,
Already I'm lost.
yeah, we'll try to pull this apart. We'll go slowly. We're not gonna, and as usual, we're not gonna do sort of a heavy math focus here. But I think we'll try to run through are sort of the concepts here, sort of the state of the art, some of the emerging technologies, how mathematical or quantitative approaches might really help to navigate some of the big data sort of possibilities that relate to this new technology in spatial genomics. And then how, I think for me, maybe one of the take homes or the punchlines to put it out early is that, this is all sort of like cutting edge, like emerging technology and emerging ways or modeling of these processes, which are in and of themselves really complex. And so, maybe with a little bit of echo of our last TWiEVO 88, we're not going to, I think, solve some major mystery of biology here, but it's sort of setting the table, I think, for the road ahead and the possibilities ahead in really exciting ways. So, before we really dig in,
Now, you don't do any
yeah,
cancer evolution yourself in your
no,
laboratory,
I
right?
don't. And so, yeah, so this is like,
Okay.
from a non-expert here. And if anyone out there has more experience in sort of tumor diversity and that kind of biology, yeah, feel free to chime in throughout the episode on the chat and correct the record as needed or point out sort of maybe major holes here, which I'm sure there'll be a few. But so we'll get into a little bit of the motivation for why we're talking about this on TWiEVO and using evolutionary or state dependent, what that means, and in terms of an evolutionary model for thinking about how tumors grow and develop or evolve. And so, and I think that those are all topics to unpack a little bit and think about the concepts there. So before we get there, let the authors. So who put this work together? I think a really impressive group. So this is Maya Lewinson, first author, and one of the corresponding authors, Trevor Bedford, who has crossed sort of some thresholds in TWiV space with a lot of the genomic epidemiology that was really sort of on the front lines of the SARS-2 pandemic most recently. He also, Trevor's lab, has really pioneered a lot of the work in genomic epidemiology and influenza as well, and then has, I think, a growing or also sort of a pillar in cancer biology as well. Nicola Mueller, who's another postdoc, I believe, in the Bedford lab, and then Allison Feeder, who's the senior author here. Allison is an assistant professor in genome sciences at the University of Washington. So all kind of neighbors here in Seattle that came together for this paper. And Allison Feeder's lab, by the way, really worth keeping an eye on. I think some really exciting work thinking about how spatial structure really influences evolutionary processes, as we'll dig into today with the tumor biology, but also for infections, both sort of in populations, but even within sort of individual hosts, and how there could be sort of spatial organization that could have a real impact on how populations of viruses and other microbes evolve in real time.
Right.
So I think,
do you know Trevor reasonably well, Trevor Bedford?
Yeah, I'm trying to remember. I think we might have met once or twice, but we haven't. So I did my postdoc, as you know, at the Fred Hutch
right.
before Trevor arrived, though, so we didn't actually overlap there. I'm a big fan, and of course, have followed sort of breathlessly the Twitter long-running Twitter feeds to sort of understand what's the diversity or where is SARS-2, the variants sort of emerging from, and how to sort of unpack that But we haven't spent a sort of big fan, I guess, is how I would describe my
Maybe
relationship.
I'd love to have him on TWiEVO. It'd be good.
Oh, great idea. Yeah. Yeah. I'll keep an eye open for some work that might make sense so we can invite him over. Okay. So I think, to begin with, a core conceptual question here is, do tumors really evolve? So when we think about evolution and what that is, generally there's sort of a generational component of it, or there's a vertical inheritance involved, right? So I think we're all comfortable with the notion that evolution happens over long periods of time. Tumors, most of them, not all of them, and we've talked about this before on TWiEVO, are sort of a dead end evolutionarily, right? So it might arise in an individual host, change very rapidly with maybe some overlapping echoes of evolutionary processes that happen very quickly with populations of microbes, overwhelm the host in some cases, or whatever the course is. Generally it's a somatic process, doesn't get into the germline or get passed to the next generation. So I think the evolutionary purists might have a bone to pick about using that term evolution. I think most of the field has gotten over that, and they just say, yeah, tumors evolve, there's genetic variation, this happens. Some of the exceptions out there that maybe are closer to sort of classically described evolution are the infectious tumors that are contagious. And so on TWiEVO episode 46, titled "Can an Old Tumor Teach Us New Tricks?", we talked about the dog venereal tumors that transmit from one dog through to another through sexual contact. And really interesting case of genomics. Now this thing is spread around the world in dog breeds. I think there might be a little bit of controversy on sort of how you collect all the data, et cetera, but some really interesting patterns in terms of the types of mutations depending on where the populations of the dogs reside. So that's sort of maybe one of the more clear cut. There's the Tasmanian devils that fight each other and they pass tumors sort of from snout to snout, and those sort of fall maybe more cleanly sort of in a definition of evolution. Yep. Here, the authors define the evolutionary process they're talking about, which is cellular subpopulations. This is within a tumor or a collection of tumors within a host that are expanding and diversifying. And so the idea is that it's a mutational process where new phenotypes arise, and that helps the specific cells that have those mutations or phenotypes compete against others. And so certainly a lot of overlap here with sort of standard evolutionary processes. And so the idea is, can we use then evolutionary frameworks, or in this case, develop models and use simulated data to advance that, to try to capture some of the sort of core parameters or components of this sort of flavor of evolution.
So Nels, can we just start by talking a little bit about what kinds of mutations get a tumor started in the first place? Right?
Yeah, absolutely. Right. Yeah.
I mean, we understand that cells become transformed in a number of ways. They can be transformed by a virus infection, by chemical mutagenesis, by ultraviolet irradiation, by random changes, and then they start to grow. And then as the cells grow forever, they don't stop. They accumulate mutations, and then we think mutations in maybe a dozen or so genes then start to turn it into a cancer where it becomes a swelling and a mass of tissue. Is that a fair representation?
Yeah, I like that definition. And I think, so a lot of carcinogenic agents, I'd add in, obviously smoking and lung cancer is still one of the massive ones that is out there. And so those genes that you're describing, the dozen or so, or even maybe more, are a collection of both oncogenes, where if they get mutated or you change their regulation, that has sort of a growth advantage. That's where the growth advantage might come from as the cells are competing with the well-behaved, the ones that are under sort of regular growth control. And then on the flip side of the coin, the tumor suppressor genes, where mutations that knock them out might then unleash the ability of those cells, again, to sort of grow in ways that are ultimately not, that don't favor the health or the life of the host organism in that case.
I mean, P53 is a big one, right? Because this is a crucial detector of DNA breaks, double-stranded DNA breaks, and so forth, and I think is mutated in many tumors, right?
Exactly right. And also you brought up the participation of viruses. I think that's a really important component here. And so when we have these collisions with virus populations, as they're taking over or infecting a cell, there can be direct or indirect ways that they alter or mutate even, the host cell, or alter its biology in a way that it can become oncogenic. And so, some of the foundational work with some of the retroviruses, Rous sarcoma virus, that sort of pointed to the definition of an oncogene, or HPV, human papillomavirus, which has oncogenic potential, which is sort of on the rise. Another case, an emerging case where actually a vaccine can be super effective to block the ability of this. And so that's, sort of coming online, or as part of course, the complicated conversation there.
One of my favorite lectures in my virology course is transformation and oncogenesis, which are two separate phenomena. You first have cell transformation where they begin to grow, but not every transformed cell is a cancer cell. And then those additional changes, 12 or more, have to occur, and then it becomes a cancer, where if you then injected it into a mouse, say, it would become a tumor. And I just like it because the story of Rous sarcoma virus and it's leading to our understanding of the control of cell growth is just fascinating, and it just shows it took over 50 years for this to happen. It just shows how the concerted efforts of people are needed to figure out something like that. I just love it. It's great. And,
I do too.
You mentioned papillomavirus. The cool thing is when the HeLa cell genome was sequenced, HeLa cells, of course, derived from Henrietta Lack. She had a cervical tumor, which killed her, but a piece of that tumor ended up becoming HeLa cells. When the genome was sequenced many years later, we saw the papillomavirus oncogenes inserted into her chromosome, and that's what drove the cells to keep multiplying. An erroneous insertion of those viral oncogenes. And you may ask, why are they there? Because when papillomaviruses infect cells, they have to make sure that the cells are dividing. So they produce the E6 and E7 proteins, and that gets the cells dividing. And if the genes accidentally get integrated, then they keep dividing forever. And by the way, that was the subject of one of the Jeopardy questions on the TWiV 1000, the E6,
nice.
E7. I think the question,
cool.
in Jeopardy, you give an answer, and you say, this protein was responsible for Henrietta Lack's tumor. And the question is, what is E6, E7?
That's great. Yeah, very cool. That's right. And it really highlights, I think, the different sort of, as we put this into that evolutionary framework, it really highlights sort of the different evolutionary imperatives. So the virus, it's not that it wants us all to have tumors and metastatic processes unfolding. It just wants to divide. It wants to have a place where it can divide the ones that find places where they can divide, whatever that mechanism is. That's, you could, sort of conceptualize that as sort of a side effect here. But yeah, I agree with you, Vincent. I mean, one of the exciting things about the oncogenes, the discovery, the Nobel Prize to Bishop and Varmus, I think, was that, the mechanism here. So it was that somehow a host gene, ultimately the oncogene, Src kinase, so different viral kinases, somehow was moved from a host gene encoding that protein, was moved from a host genome to a virus genome. And then that has actually in turn inspired some of our work. Getting back to your point about this sort of basic science and just being curious about how genetic processes work or evolutionary processes work, that's really has been sort of fueled our research in my lab as well. Okay, so anyway, I think we think about, or I don't know about you, but for me, I sometimes, as a non-cancer biologist, just think about a lot of this as clonal. So however that tumor started, the cells in that tumor, they're mutated directly by or influenced by a virus infection, or a new mutation in P53 arose, or some combination of mutations as you described. Then, sort of the simplified view that they all just divide and it's all kind of the same thing, or a homogeneous mass of cells that just grows faster than regular tissue and then gets you into health trouble down the road. Now, what the authors here are saying is, hold on, step back. And the field is saying, and has been for decades now, is that there's a lot of heterogeneity in a tumor. So the physical location of where a cell is might influence how much it divides. And because of division is linked to mutational processes, how many mutations that lineage of cells will develop. And so pretty soon, not only do you have sort of the tumor competing against the host or competing against the cells there, but it's also competing against the cells as part of the tumor. And so you can have sort of this complex diversification or evolutionary process showing up. And so this is defined by the authors and others in the field as boundary-driven growth, that being on the outside of a tumor, located there and sort of the sphere or whatever the geometry is, is a different sort of growth potential phase than it is if you're at the center. And you can bring in all kinds of complexity. Is the tumor vascularized? How does the center of the tumor get nutrients versus the exterior of the tumor? Is there space for the cells to sort of divide and move around, et cetera? And so the hope here is to develop a model to try to capture some of this biology and to actually have a way of quantifying some of these processes to define them. And I think the ultimate hope here is that if the evolutionary framework is useful, it'll ultimately become predictive, that you might be able to say, okay, there's a high chance that you'll gain drug resistance because of sort of the patterns that you see as you compare tumor growth and see what the clinical outcomes are. It might tell you something about the process of metastasis. If you have sort of a signature, a quantitative signature, does that then predict ultimately that, in the next several months, something is about to happen? This is the time to intervene. I think those are sort of the big picture goals here.
And as the tumors are growing, they're accumulating additional mutations. That's the point of this paper, in fact, right?
Exactly, and in different ways. So the more you divide, and they point out there's an assumption here that the sort of with division will come more mutation. There are other ways that cancer cells might mutate, but that's probably one of the main ones. And so that's what their model tries to capture, is that process. And so, this differential growth, sort of the microclimates of tumors are some of the ideas here. And that this could, if you just take that sort of homogeneous view or just take a biopsy in one location in the tumor and then sort of try to apply that or as a veneer across all of the biology, that you'll miss a lot of the sort of biology that's sort of unfolding right before your eyes. And in particular, there's, if you put this then into sort of a population genetics sort of view, there's this what's called the neutral variant allele frequency, or the spectrum, the VAF spectrum. And if you just sort of take an average of mutations across the whole tumor, you might sort of fool yourself into thinking the mutational process is really different than if you highlight or take the sort of the spatial locations into account.
Now, Nels, when we get sequences, do we excise tumors? Do we do biopsies or all of the above? Is that how? And we never get every cell in a tumor, right?
No, but sort of this is where I think that's a great question, because it's really interesting how the landscape of the technology is sort of changing in parallel in real time. And so, I think, and we'll see this as we delve deeper into the paper, so they actually look at some an actual tumor data sets. And here, I think I might be getting a few of the details wrong, but the big idea is that they remove the tumors as part of the treatment process, and they do what's called punch biopsies. So they take little collections of cells from different locations and actually map where they're taking it. So they take some from the center, they take some from the exterior, because there are all of these ideas of boundary-driven growth. And then we're sort of entering, on the technology front, the genomics front, we're entering sort of this era of spatial genomics. There's entire companies, one in particular, 10x Genomics, that's probably sort of the farthest along. They have these like remarkably expensive analysis pipelines where whether it's a tumor or a developing tissue or whatever it is, you might take a section, put that on a slide. You could do that with a punch biopsy. You could do that probably with a bigger piece of a tumor, for example. And then you overlay, sort of the ability to do genomics all the way to single cell resolution. And so that would be sort of on the extreme of like collecting a lot of data from like very, in terms of microclimate, the climate is a single cell. This analysis that we'll talk about today doesn't get there. It's just, like a dozen or so punch biopsies, or they'll simulate, 50 or 100 locations. But you can kind of imagine these sort of technologies or in the sort of quantitative approaches sort of going in parallel tracks. And just as the genomics get stronger, if the models become more refined and sort of less computationally expensive, that you could imagine then, sort of tackling down to really fine-grained, high-resolution views of tumors mutating and diversifying.
And we should mention there are big cancer databases available where you can get a lot of this information, right, Nels?
Absolutely. Yeah. And a lot of published papers. I think the authors sort of do a nice job here of trying to, try to point out that, they're not the only game in town. There's a lot of groups and a lot of energy, a lot of resources are being poured into understanding and their existing models, and they'll argue for why they think a state-dependent one might add something to the conversation here, and we'll define that in a moment. but yeah, so that's really what's happening kind of in real time. And so I think it's sort of, a lot of different groups trying to say, hey, why don't we take this into account and see if this works better, or this allows us to sort of analyze this from a slightly different viewpoint. Is there value in that? Ultimately, it might be sort of all of the above, depending on the question you're interested or in the sort of translational potential that you're looking for. As some of this, I'm guessing, will move into clinical genomics as well. And you could imagine a case where this is probably already happening in some cases where, a tumor is biopsied or removed. You do spatial genomics kind of stuff, and then that might, like over the course of a couple of weeks or a month, that might inform the oncologist to say, oh, let's try this treatment or this treatment based on sort of these signals or these profiles that emerge. And so
amazing sophistication.
Yeah, amazing sophistication. Although, I would put a little grain of salt in there. A note of caution is like, the sort of classic cliche, just because you can do it, should you? Or what's the real value added here?
Sure.
If you map everything, are you really sort of prioritizing something versus if you just do sort of the traditional approaches, how much do you gain versus how much does it cost? It also raises we won't go too deep into this, it raises a whole nother collection of like ethical things. If this is available to the sort of the richest people in the world, but not sort of the global population, how do we think about global health, et cetera? So all of those, I think, things apply to sort of personalized genomics, personalized medicine as well in this space of cancer biology.
So in fact, the cancer genomes are kind of biased in that sense to people who mostly have good health care, right?
Yeah, I would say so. Certainly like the genome level sort of analysis is biased, or it's more in the basic science realm where geneticists, genomicists, cancer biologists are partnering with clinicians in order to just say, hey, can we use these tissues? Because we're just trying to learn sort of the rule book of how this is happening, less sort of clinical or translational in that case at this stage. Yep. Okay. So those are kind of the things that are swirling together here. The technology, the ability to just see what the mutational profiles or other characteristics. Is gene regulation different, not only sort of in tumor cells relative to healthy tissue, but even between tumor cells at sort of this high spatial resolution. And so the main idea here that the authors want to bring into the conversation is this notion of phylodynamics. So, and this is a little bit familiar, kind of coming from that Trevor Bedford genomic epidemiology landscape. So they've been doing phylodynamics on virus populations, not in an individual, or in some cases an individual, but mostly between individuals among populations, whether it's, influenza moving through birds, as we've discussed, or if it's on TWiEVO, or if it's, SARS-2 sort of variants cropping up and moving through the shapes of the trees, which is the relatedness of the sequences that you're sampling, actually has information in there. And so the authors are sort of porting that notion of phylodynamics. If we understand the phylogenetic processes, this can teach us something about the mutation or the sort of the growth, the birth and death of cells, as they'll describe it in the growth of a tumor. And then this also gets at, so the state-dependent evolution and the model they develop, which they call SDevo for state-dependent evo or evolution, is that they really go for this idea of boundary-driven growth. And so that's a state. So whether you're on of the cell in this case, the state is you're either on the outside of the tumor, the outer rim, so to speak, or you're in the center. Those are the states. And so then the idea would be how much of your biology will depend on the state that you're in, and can you identify those patterns sort of based on information that you can easily, or maybe not so easily, but that you can gain as the input for the model, and then you get an output here as well. And I actually think they define this pretty nicely, the inputs and outputs that they're looking at. So the input for their model is the cell state. So what's the location of it in the tumor? And to begin with, it's very simple, two states: either you're on the outside or you're on the center. And then sequencing data. And so we'll go into a little bit of the details in a moment. But so then you take, in this case from the punch biopsies, here's a set of mutations for this state, and for this sample, and here's a set of mutations for the other state. And then the model, and this is where the mathematics comes in, and we'll do we'll just kind of hold that as a black box, I think, for today's episode, and not try to sort of navigate equations, et cetera. But then, so the model takes that information as input, And then the output it gives you is these trees. The tree, and what's important here is the phylodynamics, the tree topology. How do the points, those on the tips of the branches of the tree, relate to each other in terms of where the branches connect in the shape of how they connect? And we'll dig into that a little bit. And then finally, they take the sort of shapes, and the output connects that to the timing. It tries to infer, okay, when did that cell lineage or that set of mutations sort of arise as you relate that to the others? And then you can also link that to the mutational process ultimately. And so that's sort of where the sequence then becomes linked to the phylodynamics. And potentially there's some real, I think, value there where now you start to say, okay, here's the set of driver mutations, those oncogenes that are known or new ones, et cetera. And you sort of put that together now with the collection of the population and how the cells are related to each other. And so that's sort of the new ground that they're covering or developing with this model, SDevo, mathematical model that they're deploying here for this process. So a couple of terms I think that are probably worth defining, and actually this is where I might fall a little short. So they're thinking about birth rates and death rates for the cells. The birth rate's kind of clear. I think it's when did this cell with a set of mutations that make it of a unique sort of sample or a tip on the branch of a tree, when did that emerge timing wise? And then the death rate, I don't think that's like literally the cell is like, there is birth and death of cells. As cells divide, that might be a birth event, and then cells will die. Here, I think it's more of a phylogenetic or phylodynamic definition, which is you don't have sort of the branching doesn't continue from that cell. So the cell itself that's been sampled might otherwise still be alive, but in terms of the phylodynamics, in terms of the phylogenetic profile, it would be dead in the sense that there's no more branches coming off of it down the road. It's sort of marooned into the center of the tumor where there's no space, or it's getting outcompeted by the other ones. And so we'll walk through here kind of in the next few minutes how they do the simulations to develop the model. And then at the end, they actually take some data. These are from hepatocellular carcinomas, a recently developed paper. And it's not like, so the authors are very curious of the data sets of that analysis, of doing similar analysis, although they didn't put it into that phylodynamic perspective. But so, these data sets and analysis are already out there in sort of many flavors, shapes, and forms. And then I think this group is saying, will this evolutionary framework add even more value to sort of the analysis pipeline or the kind of tools? And what they come away with, again, sort of giving the punchline here, is that for that's two tumors, that they take the sequence data, the different sampling from the exterior, the boundary-driven growth versus the center of the tumor, and they propose that their model shows that there's like a 6-time faster rate of birth at that exterior, and the other one there's a 3-time faster rate of growth, but it gets a little bit complicated, and in the end they don't have a ton of the samples are somewhat limited compared to the simulations that they can do with artificial data that they create. Okay, so that's sort of what defines our parameters of our TWiEVO 89 conversation. And so, but it's, I think, taking a few minutes here to kind of just walk through what they're doing. And the paper, it's in the journal Nature, Ecology, and Evolution. It's open access, so hopefully some of you have already dug in a little bit, or feel free to follow along at home if you're so inclined.
And by the way, folks, ask questions.
Yeah, exactly.
Slow us down, most basic question. The most basic questions, just ask. Agreed. Because this is not an easy one, and it'll help clarify for us. Now, I know you're rather quiet. There's 66 of you here, and
yeah, don't be shy. I'm a shy person.
I'll be the first to admit I don't get a lot of it.
You and me both. I'm a shy person, but there's something about live streaming on YouTube that sort of draws you out, which I can't. I'm just really enjoying this. So I'm happy to sort of drag us through the weeds here, but absolutely, ask questions, and let's see if we can sort of kick this around a little bit. Okay, so they start with what looks like a really simple kind of simulation, which is to basically set up a 2D lattice, two-dimensional lattice in silico. And if you look at figure 1, panel A, you can see sort of a visual representation of this. And so it's just sort of a visualization of what a tumor might look like as cells are dividing. It's color coded. From center, there's dark colors out at the edges, or light colors, and that's color coded. Great idea. Versus, which is trying to account for birth rate. So the lighter colors, the yellows, are a higher birth rate. In the middle of the tumor is darker, which is a lower birth rate, meaning fewer cell divisions here. And these are, these are just like in silico scenarios. And I think there's probability set up with, if you're basically near the edge, there's a greater probability that you'll be able to successfully give birth or divide. And if you're in the middle, it's harder to do that. And so you can, and it'll be a little bit different because it's it's a probabilistic process. And so what they do that's new is then they take all these simulations and then they put that into, here it is. Yeah. So they take, that's sort of panel A there in the left. Point 1 is the position in the tumor center.
Right.
Slow growth. Position 2 brings us out to the fast growth, the sort of leading edge or the outer rim. And then what's new here is that these time trees. So this is like, the time tree might be, we collected SARS-2 on this day, in this day, and then how does the shape or what's the phylodynamics here? Here it's, how many, what's the, where are you living in the tumor and how much have you divided? And so again, they point out sort of that 1 and 2. So 1 is from the center, and what you can see is that dot or that tip of the branch, there's not a lot of other dark cells around it. And if you trace that back, there's a really long branch until you get to the next related set. And so it's sort of just kind of sitting there, smoldering along or just hanging out there. That's in contrast to the yellow cells that are illustrated in 2, where if you go back to the nearest common ancestor, it's a really short branch. And that's because there's another cell that divided recently. And so that gets at that idea of the different shapes of the trees, right? So it's either long branches or short branches. And even more than that, there's sort of these ladder-like shapes. And so if we move to panel C there, now they're linking this to mutation accumulation. And so the yellow ones have almost this ladder topology. It's almost like, to me, it looks more like a staircase than a ladder, where you're kind of going on steps. Whereas if you have few divisions, now that first one that had that long branch in terms of the time has a really short branch in terms of the mutations, because it hasn't divided a lot. The mutations aren't accumulating. And so that's a different topology. And so there's information in that, right? And so if you just have, here they've simulated their cells, and so they can do that color kind of, coordination here to get those patterns. But in a real data set, you don't have all that information. You have like where it was sampled, and then you have the sequence. And so what they're hoping is that the shapes or the phylodynamics of the tree that come out of their modeling, the SDevo model, will then help them to like get a quantitative sense of what's going on. So the next row of the same sort of energy here, but this is now what they've changed the simulation so that the location of the tumor doesn't matter. So this is just, you're just as likely to divide a new cell wherever your location is in the tumor. And you can see that now it doesn't have that same sort of spatial pattern, and that changes the shape of the trees. And so the phylodynamics is very different. And so that's sort of the comparison point. And so, in real life, in a complicated tumor, probably things are going to be more complicated. Maybe you hit a jackpot mutation more close to the center of the tumor. And so that might fool you into thinking, oh, that must be out at the edge of the tumor. And so that's what they're really trying to get at with their modeling here are there quantitative ways to sort of decipher all of this and get closer to being able to understand sort of the complexity of tumor biology as they go forward. Okay. So then, maybe just a quick, since we're on the panel anyway, if we look at H and I, sorry, I'll just do it like 30 more seconds. So it's sort of the lingo here is variation in the terminal branch length. And so they're quantifying that, right? So there's the boundary driven, which is that top panel, and then the unrestricted, which is the bottom. You can see there's a huge difference in the terminal branch length. That's that rare dark colored dot on the tip of the branch. And then this also relates to clock rate. So there's different clock rates based on sort of the behavior of, in this case, the simulations. And so that, now you can put this instead of a qualitative framework, which is where you just say, oh, this is what happens on the edge, this is what happens in the middle, to say, actually, here's what the data could look like closer to something like real life.
So Nels, basically on the exterior of the tumor, we have rapid growth because there's a lot of oxygen, basically, right? And blood and bringing nutrients. And inside the tumor, less of all that because the cells are all sheltering it, right? That's part of the issue.
Exactly right. And so that doesn't, this is that 2D lattice is a very simplified version of this because it doesn't account for a lot of those complexities that can
Yeah.
arise. So when a tumor gets big enough, it'll vascularize, and then all of a sudden that interior, sort of geographically, so to speak, will be different, because all of a sudden these, the blood vessels or the vasculature will bring in all of those nutrients
Yeah. Yeah.
and sort of, but that's still probably different than the exterior, where
Right.
space
Right.
might be a different consideration, where you're crowded against your neighbors or are all kind of competing for unrestricted growth. Whereas if you're out at the exterior, depending on the location and probably the tissue type, is it bone, is it soft tissue, et cetera, that will influence sort of the ability for cells to grow and do well. And so there's a lot that goes on to this. And I think that kind of illustrates, as well as, sort of in terms of modeling, starting with something that's very tractable, in order to just sort of make a first stab or to develop the model, to get the framework together, think about the parameters, the variables, and then in the future, to kind of keep building on that. And as you sort of go back and forth between real data, patterns, shortcomings of the model, sort of, and so forth, then the hope is that you can keep sort of, you have something to build from. You've built a foundation. And so, it might be much oversimplified, et cetera, but it sort of gives you like a way to go forward. We have a question from Barb Mack.
Barb, yeah.
Which is a good one. So the cells at the outside of a tumor, are those the ones that break off and metastasize? And are they more likely to be mutants? Yeah, so that's one of the ideas, Barb, exactly. And I think that's, sort of an intuitive, simple view of how these things would work, and it would make sense. So, and maybe again, from something like a probabilistic sort of scenario, where it's like, the more times you divide, and if you're on the outside, you're more likely to divide more, the more chances that you hit some of these jackpot mutations that selection can act on. And then these things start to explore. I think the cancer biology in real life is more complicated. And, for example, if that tumor has been hanging around for, like, quietly, let's say for 10 years or something like that, it now is supplied by some blood vessels or some capillaries, then all of a sudden something in the center, it might be more of a jackpot, because you're not sampling as many mutations, but it might have access to get into the bloodstream and sort of explore more tissues. But the chances are still that would be, sort of first principles. The chances are to be less likely, just because there aren't as many mutations.
Right.
Unless, like, you can imagine all kinds of sort of hypotheticals, right? Let's say there's a clone of cells or a related pod of cells, a lineage of cells in the center that hit on a point mutation that's actually a mutator mutation. And now the mutation rate is because of a different mechanism, not just cell-based, but because it's like you're not doing DNA repair as well, then that could sort of break the rules as well. And so I think that's the hope for the model, is that as it gets more sophisticated, you can kind of step through these and identify these more complex things that sort of break the intuitive rules that you might expect.
I don't know if you can answer that one.
Yeah, probably not very well. It's a great question. This is from Tom, another one of our moderators. So the notion here that SDevo saves computational time, how does that work? And so, yeah, so this is, I think part of it is sort of, and the authors bring this up. So there's, these are both, statistically speaking, these fall into Bayesian approaches. I think what most of the field and some of the models that are out there for this kind of analysis to date use this sort of ABC approach, approximate Bayesian computing, which has some like advantages for thinking about some of the patterns and how you put that into a statistical framework. The cost there is, or one of the trade-offs is that it can be pretty computationally intensive. The SDevo model is also Bayesian based, but it's sort of a different flavor. So it's Bayesian phylodynamics, which makes a lot of sort of simplifying assumptions in terms of just the shape of the tree and how you sort of derive that information, which is computationally less expensive. And that allows them to sort of churn through 50 or 100 simulations relatively easily. I think there still are some, real, like when you're talking about even, a dozen or so data sets, but if it's whole genome data set, they actually, I think, make some compromises in terms of, instead of comparing whole genome to whole genome as they put together the trees and sort of consider from a statistical standpoint the shapes of the trees or the phylodynamics, they actually, I think they downsample to something like 25,000 mutations as opposed to the entire whole genome, to sort of try to enrich for changes that might be more signal in how the dots on the tips of the branches relate to each other in the whole tree. And that's, so all of this stuff that we're talking about, I think, is still quite computationally expensive when you think about like the amount of each dot on the branch of a tree is an entire genome that you're trying to compare to an entire genome. It's at this point, it's easier to do as a subset of single nucleotide variants. This is also, I think, this illustrates, is a great question because it sort of illustrates sort of that mismatch between the state of the art or the technology, which is you can already, get incredibly rich, massive data sets here. And then when you want to sort of build these models, how do you sort of pick out the most informative information sort of to balance the cost of trying to compare everything? And so that's definitely in play here. And so I think this is an advance, but it doesn't like solve the problem. And so that's where I think it's still, for me, for my money, it's sort of exciting and worth it to make these advances, to build that foundation, which then might sort of inspire others to say, oh, well, what if you do this? You might save some computational time, or if you, or use this other approach. It's sort of like, this is why it's great to publish stuff, get it on the bioRxiv, make it open access, so that we can discuss this even as non-experts, and to try to, sort of see what, as people hear about this, how do you, how does it sort of collide in new and exciting ways? Okay, so let's return now to a little bit of the phylodynamics here. And so, and this gets at the state dependence, which is in that title of the paper. And so, so far they've built a tree, and they've, linked that to sort of the mutation rates and how that changes the shape of the tree, that ladder-like or staircase versus the little stubby sort of branches on the tree. And so now they add on so that the notion of the birth or death or the location of the tumor, they add that to their simulated data. So they vary cell turnover rates, and they see big differences in the dynamics if they if they set it up, the simulations for boundary-driven growth, right, and identify it that way relative to the unrestricted growth. And that's, I think we'll kind of, just in the interest of time, keep moving so that that's if you want to do a deeper dive, this is illustrated in figure 2. And then, sort of the next thing in building the model is then linking this, that now that you have the states in place, is linking it to the mutations. And this is really what's, I think they put forward as sort of the new sort of secret sauce of what they're trying to do, is to connect this to, since the input are the differences of mutations, to link that to the birth or death states that also correspond to the boundaries of the tumor or the geography of the tumor. And so that's where they bring, and it's in that step actually that they sort of cut new ground. And actually figure 3, if we can put that up, Vincent, I think is a really nice illustration here.
Figure 3. Okay.
Yeah, here we go. So yeah, at the top here. So this is kind of, a graphical peek of how this thing is working. So as we mentioned, that they input cell state and sequences. And so the cell state in this case is just edge or center. It's that sort of brownish red color or the light blue. And then the sequences, right, are literally the sequences from either simulated data or from real data. And so, and we'll talk a little bit more about that. So then we hit that. So that's the input. It hits this black box, which is their model, SDevo. It takes the information, the edge center, and then and the mutations, and it sort of, that's where the computational magic happens. And then what comes out of there is that you then gain a tree topology, which are, we looked at some of those trees, and there's some below. And then the timing. So these, what's interesting about this, and this really relates to the mutation process, right? So as you build the trees and relate this back to the nodes of the tree, so there's the tips of all of the sampling that's happened here, whether it's simulated or real, gives you those tips of the branches. But then what the phylogenetics does, or the phylodynamics does, is it proposes the connections working all the way back to the root of the tree. You go back down to the trunk, and in there is a lot of information that's based on the relationship and some assumptions about, as mutations arise in different cells, if they're shared between cells, then you're more related, and then that works backwards. And so the potential advantage here is now you can really say something about the history of a tumor, not just how it looks today, but you can infer the process of how this, the cells that are part of the surface or the edge, how they relate to each other, and then based on those mutations, how have they influenced the tumor sort of biology here versus the ones in the center. And if you have that spatial information and the center ones break all the rules and are not sort of, break that topology, there's some real information in there about how you think about, just the process of tumor development going forward. Okay, so, and actually I love this, Vincent. So, they say at this point in the paper, we introduce the model SDevo. Have you ever, like, have in your illustrious publishing career, have you ever used the word, here we introduce something new?
No,
Me
no,
either. And so this is,
never.
as experimentalists, it's sort of like, here's what we saw, or here's what we
Yeah,
reveal, right? Or here's what we highlight about the biology using these
no,
tools or testing this
it's a little odd.
hypothesis.
Yeah,
It's kind of fun, right? It's sort of a different approach to science, which is you introduce something new that might be a tool
sure.
to use. Of course, others do this too. Like if you introduce a new way of sequencing genomes or whatever, that's an introduction as well. But it sort of has, I kind of like that sort of entrepreneurial energy almost that comes through there. It's sort of fun.
Couple of things here. Let's see.
Yeah.
Yeah, we asked for questions. Here we go.
Awesome.
How about, here you go, Andrew, what about waste products and dead cells? Are lymph vesicles also encouraged?
Yeah, yes. Here's where, I could try to sort of finesse an answer. My knowledge of tumor biology is so limited. This is all sort of, the armchair enthusiast versus an expert, but absolutely. Yeah. So there's all of these processes going forward. And of course, I think there's some ideas about tumor growth, right, where you sort of reach a certain size and there's almost a pause because you have an issue with, if you're sort of in the tumor's best interest, so to speak, of dealing with waste products, of dealing with of sort of the access of nutrients. And if you have nutrients, there's also waste products. And as the cells are churning through and doing sort of metabolism, Warburg effects, all of these kind of things. And so I think these are great points to bring up in terms of how simple is the model and the sort of assumptions in the simulated data sets versus what's going on in real life, and sort of measuring the distance there, which there's a lot. And so, I think you could take the view that this isn't ready for prime time in the sense that there's a lot more biology going on here. Or maybe a more generous view, which is that, well, you got to start somewhere. And hopefully, as you continue to develop or put this up against real data sets or see patterns or failures of the model, when you know some of the real sort of parameters, and including the ones that are brought up in the question, that you can start to account for that or see how that might work. But yeah, very fair question, actually. I think there's a big disconnect there currently.
Here we have one from Prid. So does it mean the health of the tissue branches determines the strength of the tumor? And if one can create a system to weaken the outside, that may interpret in killing the interior?
Yeah, that's a great question. And so I think there's a lot of therapeutics emerging now, or newer ones, where it's actually trying to break the shell from the exterior so that the immune system, right, the immune cells can sort of come in. So there's a lot going on here in terms of the growth. And so I guess getting back to the first question here, which is an important one, is that the idea here, in my interpretation, is yes. So if you have like a massive ladder-like sort of tree topology with all kinds of closely related cells from the lineages that you're sampling, that might say something about the growth of the tumor, the strength of it, in the sense that we'll see that sort of in some of the data here, where the edge of the tumor is, from the analysis of the model, is proposed to be 6 times more than right at the center, or 3 times in another. And so I think, yeah, you'd love to start to be able to sort of gauge those parameters. There might be clues about the trajectory or the strength of the tumor going forward that could ultimately influence the types of interventions that you may or may not take in terms of the side effects of chemotherapies, et cetera, or the different ways that you would attack it. Yeah. And so that's what, yeah, the time, that's the notion of the phylodynamics that gives you some time sort of predictions, the relationship of the cells, and that there's sort of clues about the biology there. Yeah.
How tumor necrosis would affect this model?
Yeah, and that gets into the sort of the life and the death, the birth and the death. And I have to say, I was a little disappointed just from as a non-expert, that I don't know that they define that. Birth seems pretty clear and intuitive in the sense that it's just when a cell divides, that's the birth of a new tumor cell. death here, whether that's truly necrosis, cell death, or it's just sort of stasis, that wasn't clear to me actually, and I didn't go deep enough. I didn't have the time to really sort of try to tease that out. That would be, I have the same question myself for the authors. That's a really good one, Lori, and I don't know the answer. Yeah.
Here's another one. So if the tumor has been hanging around for 10 years, who or how are they working on detection of the tumor?
Yeah, this is another point, and I think that it gets to sort of like the real life complexities, and again that sort of disconnect here with the sort of early development of the model versus like if you even think about the logistics of the samples that you might collect. So and the authors actually will tackle this next. We'll talk about this in a moment, but sort of in a different way, even in a more simple way. But imagine that you resect a tumor, you take out the whole tumor, you biopsy, punch biopsy, which I think is the standard in the field now. But let's say that tumor regrows from sort of the leftover cells that didn't get, sort of excised out, or ones that had already moved on to a new location or something like that. How does that relate to the original tumor? And I think, my sense, and this is sort of guesswork, is that it would be a whole nother animal. It's a whole nother like tumor process. And comparing from like tumor 1, sort of year 1 versus tumor 2, year 10, and what that looks like, you can't make a direct line across because you've sort of, it's not the same environment or the same original set of cells that sort of kicked it off. There's been a really strong bottlenecking or selection process in however the intervention to biopsy it happened. I'm guessing, I don't know. And again, as a non-expert in the field, that you could imagine cases where you do biopsies early in the life of a tumor where you don't remove it. You just take little sort of samples of the cells, do the spatial genomics. And then in that case, the 10-year comparison to, 2-year, 5-year, 10-year comparison might actually give you a little more of that progression if if the intervention doesn't sort of impact in a drastic way the quote unquote life of that tumor. So yeah, great question.
How about this one, Nels?
Can a brain tumor have thoughts? Whew, this is great. So, I kind of wonder about that, maybe not about brain tumors, but about organoids, brain organoids, right? That you can now, which are probably take on some like tumor-like characteristics in terms of getting, if you take primary neurons and sort of put them together in the right growth conditions, you can get these little mini brain sort of organoid versions. Do those ever have thoughts? Sort of like maybe a parallel sort of, science fiction or nightmare fuel along with artificial intelligence. Imagine putting like the chat bot into a brain organoid and sort of there's feedback there. I don't know how to get there, Rob. Interesting question, philosophical question there.
It's not clear to me if they consider the computational speedup of SDevo is against other statistical methods or the 2D lattice simulation.
Yeah, this is a great question too, Nicholas. So I think, so certainly for simulating the data sets, my impression is that's not the computationally intensive step here. Like making those data sets is relatively easy. And so the reason for doing a 2D one is just to kind of get started. And I think they're really kind of leaning in on those two states, which is again, a massive simplification, exterior center. And they do say, so, again, they develop the model and then they say, I think near the end, as they do more simulations, instead of the 2D lattice, they do something, it has a name actually of how people are now simulating more, just doing in the simulations of the types of data sets that you might get. There's more sophisticated simulation pipelines and they use that. And that's certainly, I think, moving into the future. I think that really the computational speed up in the model itself or the cost of this comes into sort of that figure we showed. So you input all of the states and then link that to the sequence. And then it's in that churn of the computation to organize that data as a tree, a phylogenetic tree, and then linking that or showing the tree both as a time tree based on, where the location is, how many divisions you infer, and then linking that to the mutations and how that sort of those topologies give you information. That, I think, is the really computationally intensive part. And, the Bayesian trees or the Bayesian approaches to phylogenetics gives you some flexibility and has been used just in regular phylogenetics, right? When you're just looking at the relationship between different species based on genome data. The Bayesian approaches give you some advantages there, but very quickly as you get to the, hundreds of cells, thousands of cells, whole genomes, then this becomes like a massive crunch even with these methods. Yeah. So next one from Lori. So when studying cells in vitro, this evolution is not going to occur. But cells do change in culture over time. Absolutely, they do. I mean, that was one of the interesting things when Vincent brought up at the top here, the HeLa cells, and the fact that you've got genes from human papillomavirus literally integrated into the genomes. I remember, like sort of the mythology of the HeLa cell was that, oh yeah, we've been culturing this like cells from this massively aggressive tumor that Henrietta Lacks ultimately succumbed to relatively quickly. People said, wow, those genomes are going to be a mess. There's been, people for 50 years have passaged these cells in totally different lab conditions. You've got, the idea that the chromosomes are going to be scrambled. There's going to be massive mutation processes. That was sort of one of the, so a really interesting story from a few years ago. Like there were two groups, one at the NIH, if I'm remembering right, and one in a different lab in Seattle in genome sciences, Jay Shinduri's lab, that did the sequencing of the HeLa genome. And I think it was surprisingly organized. I mean, like you're saying, Vincent, you could pull out the papillomavirus E6 gene.
Oh yeah.
And some other, I think there's like,
I think they're both integrated into chromosome 18, I believe. Doesn't really matter. But
yeah. And there are some, there's some aneuploidies. So like some chromosome numbers are a little bit messed up, but it's not sort of like just a total mess. And it's somewhat consistent even between different HeLa lines. There is some real controversy around doing that genome without the permission of the family. And so, really interesting genetic and ethical conversation there that overlaps into health ethics as well. I guess the long story short is that in vitro, very different than the tumor biology, I would say. And so I think there's like and sort of the strength of some of this new technology is potentially that you can really go to the tumor in situ or pull it out and sort of do real-time analysis. What had limited us in the past was that what would push you towards more in vitro systems for understanding cancer genomes or cancer genomics is to have enough material to get enough nucleic acids to measure like what are the mutations or to measure what's the difference in transcripts, the gene regulation that's going on. And so those are some of the compromises that you might take it out of context and grow a lot of the cells in order to get enough material to just do the sequencing. What's sort of the advantage here and the hope is that because you can get like even with a single cell, we can now squeeze out enough sequence to have, and this is ongoing technology development, but to have high quality data sets, even from single cells. Now that sort of opens up the possibility of actually going to the real biological process and seeing it unfold. And that's really, that's happening on multiple fronts, tumor biology, cancer biology fronts, the genomics, the genetics and genomics front. And then here, sort of the statistical evolutionists are coming in and saying, the population geneticists are coming in and say, hey, we can add something to the conversation too, which is modeling the dynamics of these from a genetic standpoint. Yeah, great. These are great questions.
There's some more. So we got, Lee says, when I was an OR nurse, there was talk about if you biopsy a tumor, would that spread cells in the patient? Not sure where the science is on that. That was an issue with morcellation of uterus.
Interesting. Yeah, I can't answer this one, but I agree. Like how you, that certainly could change, potentially, like you could imagine, at least as a non-expert, that if you biopsy something now, what it was originally in the center is now there's access to, at least in the short term, depending on how invasive that is, that could change the tumor dynamics. It's a really good point. The other thing that's an important point here is actually the sort of the compromises, which actually I think builds very nicely from these last two questions, is that the punch biopsy isn't just taking single tumor cells. So each dot at the end of the branch of the tree here is not a single cell. It's basically a punch biopsy. And so because of that, the authors, really spend a lot of time considering that there could be some biases in the data that you recover, where you're sort of, you're not making you're not treating the whole tumor as homogeneous, but you're treating the biopsy, the micro punch biopsy as homogeneous. Yeah. And so they actually try to simulate that. So they say, like worst case scenario, if there are center cells and edge cells mixed together, would that sort of how would that cloud the data or sort of throw sand in the gears of the ability of the model to return patterns that are informative. And so I think the answer there, and again, just in the interest of time, I think we won't go way into the weeds, but the answer is that that could be an issue. And so as they continue to develop the model, they're kind of getting a sense of what those patterns would look like when you sort of don't have ideal sampling. And that's, of course, getting to the last question from Liz. I mean, that's like, the first priority, of course, is the patient. And the doctors are making decisions about how they're treating the patient, not based on, is this going to be a great data set for genomicists to pour over and develop models. It's how do we have the best outcome possible with the sort of the state of the art or the available resources to make medical decisions. And so, that's, and so that's, I think that's an important part of the process of building this model or taking this quantitative approach, is realizing sort of the limitations of the samples that might be available or are available, already published, or might be coming online, and sort of adding that to the conversation and in the model development. And so, that's sort of in the next figure or two. They trying to simulate not just sort of a tumor as a whole with the different sort of types of boundary-driven growth, but
figure 4.
Exactly, figure 4. They also take into account where cell samples are taken from the tumor. And they simulate the punch style biopsy as well, and see how that influences it. And so actually, so here, if we just go along some of the estimated birth rate differences versus the true rate differences, they're hoping that these match, right? That they don't fall off of the line as you go, that the true rate difference actually mimics or is captured by the estimated birth rate. And so you can see, like in panel A, that looks pretty clean. There's the dotted line, the dots, there's some noise there, but the dots generally follow suit on the y-axis and the x-axis and the y-axis. B is worse, so especially at that true birth rate difference that's lower, which they know because they've simulated the data here. The estimated doesn't return as sort of clean of results. And so they sort of march through this. I think if you scroll down a little bit, Vincent and D, E, and F. So here now they're doing... Yeah, no, you're good. So here they're doing a couple of things. So one is they've moved from that sort of simple 2D to this PhysiaCell. That's the sort of more kind of complicated simulation here, going from 2D to 3D. And then the reason you have those, now those collections of dots through time is that they're trying to simulate the biopsy, where you take it, you're taking different... the samples are now collected like at the, from the biopsy, and so they come out as if they're clustered in time, and so, or clustered in birth rate, and so they're still trying to get a sense of whether that follows the dotted line, and the answer is not perfect, but it's pretty close. Looks like there's differences too in the estimated birth rates that are sort of underestimated when the true birth rate is low, and a bit overestimated when the true birth rate is high. You can see that maybe in particular in panel D there. So so that's sort of that I would say concludes the simulation and the development of the model that they introduced, SDevo. And then in the last piece of the paper, which is probably the most interesting in a sense, is like, okay, what if we try to apply this now to real data sets? And so they move to this published data set. So this is these were 3D spatial microbiopsies followed by whole genome sequencing, and published by another group. And here's an example, I would say, where making the data publicly available is massively useful, because now other scientists can kind of pick it up and play with the data, use it in new ways, develop new things. And so I would say, in the last decade or so, this continues to be very contentious. It's sort of like, well, the authors of the original paper have put their blood, sweat, tears, limited resources into collecting these data sets. And as soon as they make it available, the sort of dumpster divers, like my lab included, we can just come in and, sort of without paying a dime, have access to this data, sort of crunch it in a new way, take credit for our fancy new way of analyzing it. And then the original group is like, well, hey, what about us? And so that can cause these incentives to try to keep your data hidden, or your most interesting data hidden. I think we're now, enough folks in the community are coming around to the idea that making science open, the more open access, making code available at GitHub for you can pick up the SDevo model and develop that yourself, et cetera, is becoming sort of a little more the rule than the exception. And in fact, a lot of journals are now requiring, if you want to publish that, or NIH is requiring, if you want to be funded by us or continue to be, that the data that you collect with taxpayers' money should be available to the wide world, so other people can really dig in and sort of bring their own development to this to unlock the potential of these data sets. There are some real trade-offs here, right? Like the incentives to put in your limited resources to collect it in the first place. Anyway, I'm onto a diversion as usual. The point here is because this is available, the authors can take this. Maybe, Vincent, if you put up figure 5, this actually I think is really a nice illustration of what does a 3D spatial microbiopsy mean. So here's tumor 1 at the top left, obviously simplifying it into a sphere, but they're taking the samples that were collected and published and whole genome sequenced are from each of these locations on the tumor. So we've got and then the authors here have color coded it. So what they consider an edge state versus a center state. And the light blue ones were from more of the middle of the tumor, whereas the sort of brick red ones are from the edges and at the sort of top of the tumor for like those 3 Z samples. And then what you can see now is with the real data is in panel B is the genetic tree that forms now 2 state or color coded for the state. And then the inferred tree time. So in C, this is their model. This is what SDevo does. And then in D, this is moving from that sort of qualitative. Oh yeah. So in panel B, the genetic trees, which the authors, since they've whole genome sequenced, they've already done that analysis. So they say, okay, the center ones are more related to each other. There's fewer mutations, and that's important here. So the scale, that little scale bar, 10,000 mutations per, that gives you a sense of the difference between the samples from the whole genome sequencing. And you can see that the light blue ones, the center ones, are actually, there's very little distance between them. And they're drawn just sort of vertically. This is that center where it's basically fewer mutations accumulating that what you would expect from some of the assumptions here. Longer branches for the ones on the edge. And then so C, this is what's new from the SDevo model is that now you get the inferred time trees. And I think, they're a little, and then because it's quantitative, you can then now do birth rate estimates. This is where they get on the very far right there, they think the cells at the edge have estimated birth rates, roughly 6 times more with confidence intervals compared to the ones in the center. And that comes out of the estimated birth rate analysis. I think what they were really hoping was that their inferred time trees would have a little more of the character that we saw in that simulated data, that ladder-like or stair-like sort of features. It kind of doesn't. It's sort of, there's this same as it's kind of pretty close to the snapshot that you get from the genetic trees. The time trees don't add a lot more information in this case. And so I think that's where the authors, I think, nicely sort of point out, the limitation of the model so far, or gives you a sense of the data sets they might need to see that. And so they think that, there's just not enough sampling here, actually. You have to get to, I think there's, what is this, 10 samples, 12 samples, something like that.
Yeah.
Something closer to the 50 or 100 might be what you require to really make the model sort of reveal the phylodynamic patterns that could give you some more information about the types of evolutionary processes that are playing out. So it's tumor 2, kind of an independent comparative data point, which is really useful. A little less sampling, some of the same, I think, broad strokes in terms of the genetic view and the time tree kind of are overlaying here. Here, based on the data they have, instead of 6x, like the estimated birth rate came out at 3x quantitatively. Again, sort of limited data. You don't see all this. What's interesting here and kind of hiding in panel G is that some of those, if we go back from the tips of the branches to some of the more ancestral nodes, the trunk of the tree, so to speak, there's actually some of those brick red dots up there, hanging out up there in the center samples. And so does this, what's going on there? Is this because of the biopsy, some outcome there? Is it just that the model's getting confused? There's some more complexity hiding there. And so I think it's sort of like, maybe an analogy we could draw here for looking at this real life data. It's sort of like test driving a car. And at the end of the test drive, you're like, okay, the car moves. It rolled out of the parking lot, but it wasn't like a, fancy new electric vehicle that was going 0 to 60 in 2 seconds. It didn't sort of blow the doors off so far. And so yeah, so that's it. So I think that's, that kind of gets to the conclusion of the paper in terms of, developing the models for showing us the state of the art, a little putting this, and it's sort of like maybe the test drive outcome was like, okay, we were driving around in a Ford Pinto instead of a Ferrari or something like that, for the older crowd out there that knows what a Ford Pinto is. But so that's
what would be next? Would would they get more data and validate the model further? Would that be the idea?
I think so. Maybe a great outcome would be like if a cancer biologist read that paper and was like, oh my God, actually we're developing the perfect data set with this population of patients, maybe partnering with the group, having those conversations between all the way from the clinic to the sort of basic science population, biostatistical nerd energy, like all the way through, right? I think that's one of the potential outcomes. This is where they really highlight, one of the strengths here. This I think builds off of Tom's question from before, which is the advantages here. So, the car is not running well, but does that Ford potential have the potential to become a Ferrari?
Yeah.
And they would say it does, because the fundamental difference here is that it's not the ABC approximate Bayesian computation that has some probably, sort of real limitations. It's this, Bayesian phylodynamics, which can probably, really move forward or can continue to develop as they continue the work. And so, the other, and the other point that they make here, and I think it's an important one too, is that sort of the application or the test case that they've obviously lifted up and developed here in the simulations and in the real data sets is the cancer biology. But are there, boundary driven effects or different states that actually apply to a lot of different biology? It could be an infection in a population. I think it could even be an infection in a host. So Alison Feeder's group also has sort of a whole nother sort of line of research that involves thinking about the evolution of viruses within a host, and some of the ideas that we've talked about in terms of like an immunocompromised patient with a chronic infection of SARS-2, or another more long running pandemic, an HIV patient that's on triple cocktail therapy for a decade, and thinking about the cocktail therapy for HIV has been massively successful, but there's probably times where that, like whatever drug you're taking, you forget to take the drug, or that drug, like this action works really well in one set of tissues, the lymph nodes or something, and much less in another tissue, the lungs or something like that, or a different set of immune cells. And so does evolution play out differently for the viruses that see different levels of drug depending on where they're replicating or the types of cells are replicating it? And so these models, the SDevo model, and that approach of phylodynamics could be applied in other settings as well, not just the tumor biology. And so I think that's sort of the direction that the work is going into the future as well.
Okay, off topic. What's the dolphin? Yeah,
there's a dolphin on my whiteboard?
Some kind of a fish. Maybe it's not a dolphin.
Oh, yeah, that's a zebrafish. Let's see, what is it saying on it? I can't even remember what I was trying to illustrate there. It's been sitting up there. We've talked about this. I think on TWiEVO, we had a fun conversation about virus discovery in zebrafish. So we discovered the first natural coronavirus of zebrafish, probably the first natural virus in some sense. With another group. And so we're continuing that work. We're doing more virus discovery in zebrafish. We're thinking about the genetic tools. And so that's what that dolphin back there is a sort of sad illustration of a zebrafish.
Okay, Tom says, I've read of tumor stem cells. Are we looking at emergence of novel populations, or do they remain a special population?
That was a great question. And that was the notion of a tumor stem cell, and gets at some of the cell lineage. I think the phylodynamics might actually be a really useful approach for trying to quantify that. And so, does that still hold up in tumor biology as a useful concept? I think the answer is in some cases yes, but in others maybe, I don't know if that sort of trend is sort of peaked and is coming down. I'm going to punt on this one just because I don't follow the field close enough. But that would be something closer to a stem cell would basically be a cell that would in that phylodynamics, it would, it might be ladder-like, it might be, there's sort of contributing to all these recent cell divisions and yet retains some sort of ancestral state. That would be the idea, that would kind of keep contributing. Moving, excuse me, this cold is catching up to me, moving forward. And so I don't know the answer to that, but I think it's possible that some of those dynamics or something like that would come out of this kind of analysis, and that would alter the topology of the tree, which is what sort of phylodynamics is all about.
So Nicola says, yep, seems that the big speed up is from using Monte Carlo methods to evaluate the big integral that updates the probability distribution.
Exactly right. Yep.
Okay. Let's see. When dividing up a tumor to go to different research labs, could you end up with two very different cell lines?
Yeah, interesting. So I think the answer is yes, especially given time. So in this case, maybe this illustrates that sort of doing the data analysis as close to in situ with as little sort of ongoing manipulation as possible. And so, yeah, but imagine like, let's say a cancer biology lab has a tumor, they derive a cell line from that, and then they share that with 10 other labs, and then come back 10 years later and ask how different are those cell lines from each other. And so it's like these things will be propagated in different environments. So they might be frozen down in liquid nitrogen. One group might get them to divide, like hundreds of generations and constant sort of churn. And there's a passage number where a lot of changes have occurred or potential changes have occurred. Another lab might, like thaw it out, run it for a year, put it back on ice, and then bring it out 5 years later. That would be a very different sort of tempo of the number of cell divisions, which is definitely going to influence it. Even cell culture conditions are going to influence this. So if someone's serum, sort of the nutritional broth that these things are grown in, is from a different batch or a whole different company, that might influence the mutations that get selected on as they appear. And even the number of cells, like how long are they allowed to sort of cohabitate together in sort of a confluent monolayer versus, are they in log growth versus stationary? All of these things could contribute, and you could end up with very different cell lines.
All right, let's do one more, Nels, and then we'll do some picks because you're
running out of gas.
Running out of gas. It's okay. Right. Tom's public data sets, very important. Could shared authorships, collaborations emerge from the dumpster diving, Nels' term?
Yeah. So I'm a dumpster diver. I love dumpster diving. So getting back to that dolphin on the board behind me, when we discovered the sequence of the first coronavirus, first natural virus of zebrafish, we could, as soon as we had that foothold, we knew what the sequence looked like. We were like in business because for 40 years, developmental biologists have deposited different data sets from different tissues of zebrafish, et cetera. And so this is like virology on steroids in the computer. It's not real virology, but this is the dumpster diving, is we could immediately, do a search of these publicly available data sets. We can make predictions about this virus. We saw most of the virus reads in intestines and not in the testes or the eggs of the data sets that were collected. We saw a little bit in the bloodstream. We could so immediately say this looks like an enterovirus because of the intestinal signal and some of the sort of patterns like that. We could also very quickly say this virus exists around the globe. Every single data set that was deposited, we could see evidence of that virus. This like actually scared a lot of developmental biologists at first, the fact that there's a virus hiding out there and that's influencing our data analysis, et cetera. But yeah, so this, for us, it was like we went from here, a discovery of a virus, to here, where we could do this sort of in silico style virology and make, advance our hypotheses and our ideas about how this thing might transmit really quickly, or how the sort of the life cycle or replication style of this virus. And so, yeah, and this can work in a lot of directions. So as soon as we published this paper, we made all of our information available. Then immediately we started getting emails from labs around the world. A lab from France emailed us and said, hey, thanks for depositing your sequence. We ran that against our fish samples that weren't or might have been publicly available. We didn't run into it. They found a variant, or actually a different strain of that virus that was about 85% identical in its genome. And so we don't think it's the same virus, but a different strain. I mean, that's sort of a delicate definition. But so immediately, like that, kind of got them onto that idea. And now they have a virus. They discovered the second coronavirus of zebrafish. Other labs came to us and they said, we've got this mutant strain, and it also looks sick. We're suspicious that there might be more going on there. Would you be interested in looking at our data sets? The answer is yes. Thank you. And let's collaborate. Let's look at this. So we've had, conversations, and we discovered a pox virus of zebrafish that appears to replicate almost exclusively in the jaws, and it shows up in the scales of zebrafish. And so this is the magic. Yeah, we're working on the genome now. We're trying to get it all together. But this is sort of the magic of open science, or of collaborating or having the conversations. You never know who's going to sort of show up and say, hey, what about this? And so a lab that's thinking about bone development and has mutants in New York will write to a lab in Salt Lake City and say, hey, we got to check this out. And we've got another discovery on our hands. It's one of the projects I'm, most excited about. And so, there's some real advantages. There's also, I don't want to make it sound like there aren't.
It's cool.
I also understand that people who have spent most of their resources to develop maybe over years these data sets and are really hoping to have the first crack at it to say, here's the payoff from not just collecting the data but analyzing it, because then of course they're hoping to publish some papers, which means there's been productivity, so the investment that was made in them has paid off. I think as taxpayers, as interested parties, as grant reviewers, we need to take a generous view and say, incentivize, if you share this with the world, the good things that can happen, and the motion, that should be rewarded anytime that's put out there, right? There's already value in that, even if you haven't sort of delineated the exact sort of conceptual advance or whatever. That should score major points for continuing to have a funding stream. You've developed something that has a lot of worth to the community. And so I think there's a little mismatch there right now in how we value that. And so people get a little bit cautious about sharing. They don't want to get scooped or have their really cool data set. Someone else gets to tell the cool story, and they lose that sort of moment. And so, fair enough. But so can we sort of rebuild the system? And let's go in that direction. A lot of groups are now bioarchive, right? People used to say even 10 years ago,
sure,
there's no way I would put this out there. Someone's going to copy and paste my research and in 2 months get it published. And in reality, it's sort of been the reverse. As more people have shared, the more it's almost like, oh, this person already had this idea. And so it's really helped. The openness has really helped move science forward.
So we have two new mods who have come, not new, but two mods have joined us. Les, thank you for joining us. And Steph, thank you for joining us as well.
Great.
Appreciate that.
Great.
All right, Nels, let's do some picks of the week.
Let's do that. Yeah. So
Before your voice is completely gone, right?
That's right. I'm just trying to illustrate the importance of infectious microbes, not just scientifically, but personally. This really influences everything we do.
You have a viral cold, I presume, right? Of some kind. Have you had a diagnostic or anything?
I haven't. So this is like the daycare pipeline. So it's sort of like, your science, sort of you replicate your science in real life. So we've been speaking of, again, that sort of dolphin behind me, zebrafish. We go to pet stores, we collect pet store fish, we put them with our lab strains, and we transmit viruses to the lab strains to get new ones. I do this in real life. My daughter goes to a daycare. It's like being at a pet store. There's all these collisions and germs and everything else. Then she comes home. I'm the lab strain.
Of course.
An old guy who hasn't been around a lot of germs recently with the pandemic. And as soon as she comes home, that snotty nose just immediately gets sort of, somehow enriched in my facial hair. And then I think this is actually maybe was bacterial, at least in part, got into my vocal cords and the sort of bright green phlegm here. I'm going to stop there in terms of the details, but that's what we're up against. The latest chapter in human virus transmission from the daycare. Okay, but so before my voice goes completely, I wanted to move into my pick of the week. This is already previewed by our opening conversation, which is I wanted to just take a moment again to celebrate TWiV 1000 and congratulate you personally, Vincent, for really a remarkable track record of starting something with a community of scientists, a view that has been, TWiV alone would be a great achievement. I think the way that you've built this into sort of all of these channels, all these podcasts is, really unique and impressive. And so I think we're all waiting to celebrate with you when the actual episode drops and posts. And so is that going to happen in the next week? Can you give us a hint of when we might see this, or is it still too early to tell?
No, it'll be out by the weekend. Yeah.
Fantastic.
My video guy Ray is working on it, and it's just a complicated edit, right? It's 4 cameras and 9 microphones. So takes a while
Absolutely.
to get.
And I think
So
worth it to kind of have that,
yeah,
that sort of
it'll be worth it.
remembrance.
I think Ray's a
Yeah,
good editor, so I think it's going
agree.
to be fine. So look this weekend to check it out. TWiV 1000. Yeah, it's very cool.
Yeah, very
I'm
cool.
and thank you for picking it. I was going to pick it too now.
Yeah, I made a preemptive strike.
Yeah, indeed.
No, that's great. It's great, Vincent. And I do have a link to Parasites Without Borders. It sort of previews, has a picture or two. I've seen on a couple of the social media channels some of those great photos you mentioned from that fellow who's a street photographer, and the black and white sort of bringing out the hosts and the audience in the background. You could really see the energy there. And I think visual representation of what we're trying to do here and connect in new ways with our colleagues, other scientists, but also with the general public. Really effective and exciting, innovative work. And so that's my science pick of the week is TWiV 1000. How about you, Vincent? What's your science pick of the week?
I have an article in Goats and Soda, NPR publication by Jason Bobine. It's called The Dream of Wiping Out Polio Might Need a Rethink. And this is about, what I've been saying for a while is that we have gotten rid of most wild polios. Types 2 and 3 are eradicated. Type 1, very few cases a year. But we've replaced them with vaccine-derived strains. Right. And it's not clear how to get rid of those because you keep using the oral polio vaccine and reintroducing them. They made a new OPV2, which was not supposed to cause polio, but it does, even though at a lower rate. It's not clear if you can stop it from circulating. So, as this campaign has progressed, we've had different issues arise. So I'm glad to see that Amy and I aren't the only ones talking about this, and this is reaching the mass media as well, or the press at least. So check that out; it's really good.
Yeah, I agree. And so just taking a quick glance at it, exactly that sort of the patterns are illustrated here in a graph where it's the wild polio cases versus the vaccine-derived cases. And it looks like in the last few years, like the wild cases are basically gone, but it's the growth of those vaccine cases that you're mentioning that really shows you sort of quantitatively from the sampling what's going on here. Yeah, and so speaking of coronaviruses, so there's other enteroviruses that seem to be circulating around as well.
For sure.
What's your current thought on that sort of state of play?
Well, Enteral 68 is already known to cause paralysis. This is something new from 2014. Mm-hmm. In other countries, other enteroviruses are associated with paralysis. And so I think they're not going to go away. If anything, they're going to increase. Amy Rosenfeld found a cross-reactive antibody response between polio and others. So it could be as we change polio vaccines, we're changing our ability to combat some of these other enteros. So, they're here to stay. They're not going anywhere. There are millions and millions of infections a year. So who knows what can happen to them? I don't want to predict anything, but
yeah. Fair enough. Yeah, a massively successful class of viruses and sort of virus replication style. What's going on in terms, what are the prospects, I guess, for, new vaccine strategies? Does that make sense? Instead of live or attenuated vaccines, is that possible? Is there current development there? I don't think I've been following that very closely.
So a number of people modified the OPV type 2 to be less susceptible to reversion, and they introduced a number of interesting changes in the genome, like changes in the polymerase to reduce recombination and error-prone replication and some others. And the plan is to use those vaccines going forward. But I think we should have some more creative approaches, like making maybe an mRNA vaccine that you could ingest and it would go into your intestine and induce mucosal immunity without any of the replication. So I think we could work on some new approaches. I just don't think we should give up. But it's hard to find people to do that, right?
No, exactly, because of the assumption that polio is sort of in the rearview mirror. But maybe that would be best case scenario in some sense, is that because all of these other enteroviruses might be on the rise. And given, I think your point is a really important one, and a good one too, which is that some of what our own immune history might reflect what happened in the recent past with the with some of the original polio vaccines and ongoing ones. And so even if those numbers aren't high or whatever, the other enteroviruses could be, in the conversation. And so continuing to develop that technology that was so successful, or relatively successful for coronaviruses could really be a way forward. That's interesting. Yeah. All right,
Nels, that'll do it for TWiEVO 89. Show notes at microbe.tv/TWiEVO. If you want to send a question or comment, TWiEVO at microbe.tv. If you enjoy our work, we'd love your financial support, microbe.tv/contribute. Maybe we can get TWiEVO up to 1,000 one day.
Yeah, good goal. Let's do it.
That would be cool. Nels Eldies at cellvolution.org. L Earlybird on Twitter. Thank you, Nels.
Thank you, Vincent. Great to be together. And maybe a preview. I think we're going to, we're usually about a month off. I think we might move up a week or two given our travel schedule. So stay tuned for a quick turnaround on TWiEVO 90.
Cool. That is, so Nels, I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank our mods for today, Steph, Barb Mack, Andrew, Les, Tom. I think I got them all, right?
Yeah.
The other night I forgot Andrew. I felt really badly. So, as Nels says, we will be back in less than a month. But don't forget, tonight we have a live stream, 8 p.m. Eastern Q&A with A and V. So check that out as well. Thanks, everyone else, for coming today. We really appreciate your presence.
Yes.
And the music on TWiEVO, by the way, is performed by Trampled by Turtles. Let me turn that on so you can hear it. You've been listening to This
everyone.
Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious. Bye-bye, everybody.